long interspersed nuclear elements and short interspersed nuclear elements, which are also called as lines and signs. Non-LTR elements are of two types. First type is called as lines and the second type is called as signs. Lines and signs both are high copy number found in almost all the plant species. They are also widespread in most of the eukaryotic genomes. If we see that what is the structure of the lines possesses two ORF which encodes all the functions which are required for the retrotransposition. What are the functions of the ORF which are found in the lines? The first function is reverse transcriptase and the second function is endonuclease activity, in addition to a nucleic acid binding property needed to perform a ribonucleoprotein. On the other hand, if we see that what are signs? Signs, they are short interspersed nuclear elements. They usually use the machinery of the lines. Lines are of different subgroups. There are different subgroups like L1, L2, L3. They are the subgroups of the lines. Human coding L1 begins with the UTR region, that can be 5 UTR region, that includes a RNA promoter 2, two non-overlapping open reading frames which are called as ORF1 and ORF2, and it ends with UTR region. So the human coding L1 line that consists of UTR region, and then in the middle there are ORF1 and ORF2, and at the end there is another UTR region. So as there are two ORF1 and ORF2, ORF1 encodes for the RNA binding protein, while ORF2 encodes a protein having an activity of endonuclease. In case of lines, this is a typical diagram of the lines. The line consists of approximately 6 kb DNA, and we can see that there are two ORF1 and ORF2, and on both ends there are untranslated regions. The 5 UTR region of the lines that contain promoter sequences, while the 3 UTR region contains a polyadenylation signal or a poly-A tail. In case of human genome, there are approximately 500,000 lines which are present throughout the genome of the humans. On the other hand, what are short interspersed nuclear elements? They are approximately 300 base pair in size. The most common signs, they are called as ALU, and ALU, the signs in case of human genome, they are approximately 11% up to 13% in human genome. These are the typical diagrams of the lines and signs, that what is the difference between the lines and signs. In case of lines, we can see that here is a 5 UTR region, here is 3 UTR region, and in the middle there are two ORF1 and ORF2. While on the other hand, we can see that signs which are called as short interspersed nuclear elements, there is middle portion for retrotransposition. The sequence of lines and signs look like simple genes. Short interspersed elements, they are approximately 300 base pair in size, that represent reverse transcribed RNA molecules. Signs do not encode a functional reverse transcriptase protein, and it relies on other mobile elements for the transposition. In some cases, they may have their own endonuclease that will allow them to cleave their way into the genome. The most common signs in primates are called as ALU sequences. ALU elements are approximately 300 base pair in size, do not contain any coding sequence, and can be recognized by restriction enzyme ALU1. That is the reason, because they are identified, they are recognized by ALU1 enzyme. So they are called as ALU sequences. So ALU sequences are the most common type of the signs. Signs make up to 11 to 13% of human genome. So if we conclude that what are lines and what are signs, lines, they are long interspersed nuclear elements. They are 6 to 7 kb in size, while on the other hand, signs, they are short interspersed nuclear elements. They can be up to 300 base pair. And the examples of signs, the most common example of the sign is ALU repeats, which are approximately 350 base pair in size.